Hmm. See what the volume is. The volume's off, all right? Okay, can somebody confirm you can hear me?
I can hear you.
Great. I thought I had called that up, but it's not here. So let me call it up now. There we go. All right, before we begin, are there any questions? Today is June 10th, and we are on our last week of our class. Am I sharing my screen? I don't think I am.
No.
Thank you. Come on. All right. So we should be starting chapter 7 today. I'm not sure we're quite there. And then hopefully finish chapter 7 today. And then on Thursday, I will do a review for the final. Note that the worksheet for chapter 7 is due Thursday, June 12th. That's because, well, we're at the end of the term, so I want that due on Thursday instead of Saturday so I can have some time to grade it. The worksheet for chapter 7 is extra credit. Let me see if I can show that. Oops. Um, so if you do the worksheet, you'll get extra credit. I think it's 1 or 2 points of extra credit. Yeah, it'd be worth 1 or 2 points, probably 1 point of extra credit. It's simply answer 1 or 2 questions from the worksheet till midnight of Thursday. And like I said, this one is due Thursday, June 12th. You need to get it done by 11:59 and obviously 59 seconds. Any question about any of that? All right. I think we need to finish chapter 13 before we start chapter 7. Hopefully, we're close to finishing chapter 13. I have that we had talked about prions and talked about how they are proteins that actually reproduce, meaning they get a normal cellular protein, which is important in the functioning of the nerve cells, the capital PRP superscript C protein. Get this protein to unfold and then take on the forming pattern of the prion protein. And like I said, the prion protein and the normal protein are the same amino acid sequence. It's just a different folding pattern of the protein. We need to talk about plant viruses and viroid. Plant viruses resemble animal viruses in many ways. Plant viruses do cause many diseases of plants, including economically important crops such as beans, corn, sugarcane, and potatoes. I just got a message saying my internet connection is unstable. So if you don't hear something, please let me know and I will repeat what I just said. Because of the tough cell wall of plants, the plant viruses tend to enter plants through a wound or a parasite, where like a nematode will be munching on the plant, and then the virus can then enter the plant. Viruses can also enter because of fungus or where insects are eating the plant. Infected plants can then spread the virus in their pollen or their seeds so that the virus can then continue spreading. Any question about plant viruses? That's all we're going to talk about plant viruses because obviously plant viruses do not infect animals or animal cells and they do not cause human disease. Any question about any of that? And then let's talk about plant viroids. A viroid is a naked RNA molecule which causes disease. So you can think of it sort of like a naked um, virus that has only the protein code and the RNA for its genome. Only this viroid only has the RNA. It has no protein coat around it. So it's not a plant virus or a plant RNA virus. It's a viroid. All it is a naked RNA molecule, which then infects a plant cell, causing disease, including the potato spindle tuber viroid, which I need to get a picture of that. Let's see, come here, bring that in. Hopefully there's going to be a picture that shows up. Boy, this is rather slow. Oh, there we go. There are two potatoes which are infected with the plant viroid, and there's a normal potato. There's a normal potato, and there's once again uh, potatoes infected with the plant viroid. Any question about any of that? Fortunately, I should say, viroids are not something that we find that infects animal cells. They are only known to infect plant cells, and obviously they do not cause human disease because they do not infect animal cells. Any question about any of that? All right, that's it for chapter 13. Any questions about chapter 13? Let's move on to chapter 7. Always end with chapter 7 because it's a topic that students actually have a pretty good understanding of before they take the lesson in microbiology. Chapter 7 is the control of microbial growth, talking about things like sterilization, disinfection, sanitization. And students have a pretty good idea of disinfection and antisepsis already. So this would be a fairly easy topic. This slide is showing you the major goals in a rough outline. Obviously know the terms, what sterilization, disinfection, antisepsis, degerming, sanitization, autoclaving, pasteurization, what's a biocide and a germicide. And then be able to discuss the factors that influence microbial death rates. 3, be able to discuss the 3 main ways in which antimicrobial agents kill or inhibit cells. 4, understand the various types of disinfectants and how they work. And 5, understand the relative resistance of the major microbial groups to antimicrobial agents. Any questions about this slide? All right. Let me see if I can blow that up. When we're talking about microbial death, the rate of microbial death is fairly constant when you heat or treat microbes with an antimicrobial chemical, for example. And that is they tend to die at a um, constant logarithmic rate. So you have to put the log number of the microbes on the y-axis, and then the time is an arithmetic scale on the x-axis. If we were to go with an arithmetic scale on the y-axis, heard the door open. I was wondering if that was my room that was opening. All right. And you can see it doesn't follow a constant death rate. This death rate is, well, exponential, actually. Reverse exponential, but it's exponential. And if we have it on a log scale, you can see the death rate is then constant. Follows the y equals mx plus b, meaning a linear rate. So what we're talking about is when you expose microbes, and let's say we have a million survivors at time zero to some chemical antimicrobial agent, uh, only 10% of the cells will survive a minute. And then 2 minutes, only 1/100th of the cells will survive 2 minutes. 3 minutes, only 1/1000th of the cells will survive. And that's a constant death rate on a log scale. Any question about any of that? And we find that bacterial populations actually die this way. So if you have a million cells and you're treating it with a chemical and the death rate is 90% dying each minute, then you need to expose the population to at least 6 minutes time to kill off all the cells except for one. And then you could do 7 minutes to kill off that one. Any question about any of that? So bacterial populations die at a constant log rate. There's a number of factors that affect the microbial death. Obviously, the number of microbes present at the beginning will depend on the treatment. The more microbes you have, the longer time the treatment needs to be. Environmental influence can affect the microbial death rate. It's well known the presence of organic matter can affect the microbial death rate. When we're talking about hospital specimens, there will usually be organic matter around the microbes. The temperature of the treatment can also be an environmental influence which affects the death rate. Generally speaking, the higher the temperature, the quicker the microbes will die. The presence of biofilms also influences the microbial death rate. So if the microbe does not have a biofilm, they will die at the normal rate. Whereas if they have a biofilm, they will die at a slower rate. The biofilm helps protect the microbes. We'll talk a little bit about biofilms in this lesson, but a biofilm is something the microbe puts out, and then the biofilm can help protect the microbes, like mini microbes in your gum causing dental plaque and tartar. Actually, tartar is a biofilm. Any question about any of that? And it's harder to kill the microbes when they're in a biofilm than if they don't have a biofilm. The time of exposure is, of course, important. And to kill off all the population, you need to expose for a certain length of time. If there's 1,000 cells, you need to expose by about 3 minutes. This is assuming that the death rate will be 90% in 1 minute. And if there's more cells, then with the same death rate, you need to expose for a longer time. So both time of exposure and number of microbes affects the time of treatment. Time of exposure can be affected by the microbial characteristics. For example, microbes that make endospores require longer exposure to the disinfective agent than microbes that don't have endospores. Any question about any of that? And that gets down to the last property, microbial characteristics can affect the microbial death rate. We'll discuss this at the end of the lecture, but different microbes have different characteristics. And one of the hardest microbial characteristics to kill is the microbes that make endospores. If they make endospores, they will require much greater time of exposure to the antimicrobial agent than a microbe that doesn't have endospores. Any question about any of that? All right. I guess I shouldn't have shut that down yet. This table is showing you the terminology for microbial control. Sterilization refers to the destruction or removal of all forms of microbial life, including endospores. So when we sterilize something, it will have no cellular life forms within it. Now, it is possible that sterilization may not kill prions, but prions don't really have cells. Any question about any of that? Usually we sterilize things in the microbial lab by putting things in a not an incubator, a autoclave. And then we autoclave like the media, and then it's taken out of the autoclave, allowed to cool, and then it'd be available to students. So usually autoclaving is done by steam under pressure, but you could sterilize by using sterilizing gas such as ethylene oxide. Most chemical agents do not sterilize, meaning they do not remove all forms of microbial life. Like when you bleach the toilet, you're not going to kill at least most endospores. So it's not going to sterilize your toilet. You'll disinfect the toilet. And we'll talk a little bit about that. Commercial sterilization is sufficient heat treatment to kill endospores of Clostridium botulinum, and it's used in the food industry, like canned food, to kill Clostridium botulinum. Disinfection refers to the destruction of vegetative pathogens. So when we disinfect the toilet, you do not necessarily kill endospores, but you would kill all vegetative parent cells as well as any cell that does not make an endospore. So usually we disinfect with a chemical like bleach or something like that, but it could be a physical or chemical agent. Like when you boil things, you will kill all living cells. You may not kill the endospores, though. And how you would remove the endospores is what you do is you boil it. That kills the vegetative pathogens, and then you seal it, and then you boil it. Well, then you let it sit for a while, and then the endospores germinate, and then you boil it again, and that would kill the endospores that have germinated. Wouldn't kill any endospore that hadn't germinated, but if you give it sufficient time, the endospores will germinate. Antisepsis refers to the destruction of vegetative pathogens on living tissue. So that's when we apply a chemical to living tissue, like when you put tincture of iodine on your skin, that's an antiseptic. You could use bleach on your skin, and it used to be a treatment. I don't know if it's still a treatment. Bleach for putting necrotizing fasciitis, the flesh-eating bacteria. It used to be a treatment for treating flesh-eating bacteria on your skin because it was highly effective. But the trouble is, bleach is kind of harsh on your skin, but most patients would accept it because the disease was so bad. Like I said, in the old days, which was probably the 1990s. I don't know if it's still used today, but in the 1990s it was commonly used. And it was antisepsis using bleach on the skin. But when you use the same chemical on an object, then it's not called antisepsis, it's called disinfection. Any question about any of that? Usually an antiseptic is less harsh on the skin, like tincture of iodine, than a disinfectant. But like I said, bleach is a disinfectant, and you could use it on your skin, in which case it's called an antiseptic. A degerming agent is an antiseptic or disinfectant, which is fairly weak. Degerming refers to the removal of microbes from a limited area, such as the skin, around an injection site, like when you alcohol swab the skin. You reduce the number of microbes in the area. You do not remove all the microbes in the area. You reduce the number of microbes down to a safe level to then give the injection. It does not remove all of the microbes. Any question about degerming? All right. Sanitization refers to the treatment intended to lower microbial counts on eating and drinking utensils. And this is done by um, businesses when they're serving food, and it's done to make safe public health levels. It does not remove endospores. It may not even remove all microbes, but it reduces their numbers to safe levels. Any question about any of that? All right. A biocide or a germicide is a substance that kills microbes. It does not usually kill endospores, but it would kill a um, vegetative cell. We have different terms. A bactericide is an agent that kills bacteria, a fungicide, an agent that kills fungus, a viricide, an agent that inactivates viruses. So a biocide kills microbes. A bacteriostatic agent is an agent that inhibits the growth of microbes. Once you remove the bacteriostatic agent, the growth may resume. Here. One bacteriostatic antibiotic is chloramphenicol and tetracycline. These do not kill the microbes. They prevent the microbe from growing. And what we relying on, or the clinician is relying on, is the patient's immune system to fully remove the microbe. So that when you stop giving the bacteriostatic antibiotic, that the bacteria do not resume the growth. Any question about any of that? Chlorine bleach is a biocide. When you apply it to your toilet, it would not kill an endospore, but it would kill all other bacteria. Sepsis refers to microbial contamination In aseptic surgery, you remove microbial contamination. So asepsis refers to the absence of contamination by unwanted organisms. And we use aseptic surgery so that the patient doesn't get an infection from the surgery. Aseptic techniques are important in surgery to minimize contamination from the instruments, the operating personnel, and the patient themselves, as well as the air. Any question about any of that? So in surgery, usually the surgeon will scrub their hands, put on sterile gloves, use sterile instruments. The air will be filtered so that the air coming on the patient is not having microbes. And then the patient is treated with an antiseptic to remove the microbes from the patient's skin. They used to use tincture of iodine all the time in hospitals before surgery, but there are better agents now than tincture of iodine. I don't remember what the current agent is. Can somebody state what is the current agent that hospitals use to treat the patient for surgery?
Is it chlorhexidine?
It might be. Like I said, I don't remember. Chlorhexidine. It's something that works a little better than tincture of iodine. All right, let's talk about the actions of microbial controlling agents. Antimicrobial agents kill or inhibit microbes by one of three means. You can alter the membrane permeability, such as when you put holes in the plasma membrane, either damaging the plasma membrane lipids or the proteins. It will cause the contents of the cell to escape and obviously kill the microbe. So you can damage the plasma membrane. You can also damage intracellular proteins, denature the proteins. You like denaturing the enzymes, targeting disulfide bridges, like in um, what is that? Penicillin G, or hydrogen bonds can result in death, like alcohol can denature proteins, and that's one way that alcohol can kill cells, although alcohol actually dissolves the cell membrane, so that would be another way that alcohol would kill cells. And then the third way is to damage the nucleic acid. This often results in cell death. You do need to make more than one mutation, but if you make 5 or 6 mutations in a cell, you will definitely kill the cell, and if you remove an important gene, and that would kill the cells. Like X-rays and gamma rays kill cells by damaging nucleic acid. Any question about the three ways that microbial agents kill cells? All right, there are different ways you can kill cells. There are chemical agents and there are physical agents. Let's talk about some physical methods for controlling microbes. You can apply heat, you can use filtration, you can put the object at low temperature. Obviously, you can't put a person at low temperature, but if you had a fomite, you could put it at low temperature, and that would prevent microbes from growing on it. It wouldn't necessarily disinfect or sterilize the agent. You can put the agent under high pressure. You can put the agent under desiccation. You can use osmotic pressure to control microbial growth, and you can put the agent under radiation, and that would obviously control microbial growth. Oops, what did I do there? Let's talk about heat. Heat, all methods of heating, kills microbes by denaturing proteins and enzymes. Though if you have high heat, like you're flaming an object or incinerating an object, you actually turn the cells into char. and that will, of course, kill the microbe. There's different types of heat. There's wet heat and dry heat. Wet heat includes boiling, autoclaving, and steaming. And then there's dry heat, such as baking, as well as high dry heat, meaning flaming and incineration. Any question about any of that? Let's move on. Moist heat or wet heat denatures microbes by coagulating proteins and enzymes. An example of moist heat is boiling, 100 degrees Celsius, or flowing steam, which can be higher than 100 degrees Celsius. It kills vegetative bacterial pathogens, most viruses, fungi, fungal spores, within 10 minutes, but oftentimes the microbe dies much sooner. For example, E. coli, you just have to bring the temperature up to boiling and E. coli will die right away. So you just need to get it boiling and then the E. coli will be dead. So it's often much faster than 10 minutes. Endospores and some viruses do take longer to destroy. Some endospores can survive boiling water, including greater than 20 hours in boiling water. So how do we get reliable sterilization? Well, we use both moist heat and temperature in an autoclave. Using temperature and pressure, the temperature is 121 degrees Celsius usually, under pressure 15 pounds per square inch, and this will kill all organisms and endospores after about 15 minutes in the autoclave. Any question about that? So an autoclave is like a pressure cooker, steam under pressure, very effective at killing microbes. Sterilization does require the surface steam directly to contact it, but usually unless you have the bottle sealed, which you wouldn't do that because it could explode, but the steam has to get in to the agent, and that's usually not a problem. Any question about autoclaving? Pasteurization will reduce the number of spoilage microorganisms and kill all human pathogens. It uses mild heating sufficient to kill all organisms that cause spoilage, but it does not remove all microbes from the pasteurized substance. Like if you're pasteurizing milk, the milk will have a longer shelf life because the number of microbes have been reduced, but the microbes are not totally removed. The milk is not sterilized. But it will remove all human pathogens in the milk, and it will remove the spoilage microorganisms in the milk. But the thermoduric organism can survive, but they will be reduced in number, and that's why the pasteurized milk does have a shelf life. It's longer than raw milk. I think pasteurized milk can go for about 2 weeks. You can look it up because obviously there's a sell-by date, and I'm not sure when the milk was pasteurized, but it's something like 2 weeks. It's longer than 1 week because the spoilage microorganisms are not the spoilage micro, the microorganisms in the milk are reduced. There are different treatments for milk pasteurization. You can expose the milk to 63 degrees Celsius for 30 minutes, or you could expose the milk or other substance you're pasteurizing to 72 degrees Celsius for 15 seconds. Most milk in the United States, which is pasteurized, is exposed to 72 degrees Celsius for 15 seconds. Any question about any of that? Most beer in the United States is also pasteurized, probably at 72 degrees Celsius for 15 seconds, and then it's capped, which is curious because they don't have a shelf life on beer. Well, there is, but it's like several months, and it must be because the alcohol in the beer is helping preserve the beer. But there are some exceptions, like Coors beer is not pasteurized. They'll sim the beer through a filter, and so Coors beer is actually sterile. So in reality, it probably doesn't have a shelf life because it should be sterile. There shouldn't be no growth ever in it. That's something that's been sterilized properly. And if you don't know, Coors beer tastes different than most other beer. My brother really liked Coors beer because it tasted a little different. And for a long time, I used to live in the state of Oregon. The state of Oregon stated that all beer had to be pasteurized. It was a state law, and so Coors beer couldn't be sold in the state of Oregon because it wasn't pasteurized. However, the Coors beer lobbied the industry, and eventually they passed an exception and changed the law that the beer could be filter sterilized or pasteurized. And that's so now Coors beer can be sold in the state of Oregon. Any question about any of pasteurization? It's called pasteurization, by the way, because Louis Pasteur came up with the process. It was actually pasteurizing beer, what he developed it for, and that was to prevent the spoilage organisms in beer from spoiling the beer, turning the beer into acetic acid, not really vinegar because it's beer, but um, and then pasteurization was used for many other objects, including milk. Milk can also be sterilized. This is not pasteurization, it's sterilized milk by treating it at 140 degrees Celsius for around 1 second. This is called UHT or ultra high temperature milk. This milk does not require refrigeration, and you can buy it in the store. It'll be kept at room temperature, ultra high temperature milk, because it's sterile. I'm not sure if canned milk has that treatment or not, because I guess it would have to be, because otherwise microbes could grow anaerobically. So, all right. So ultra high temperature milk is sterile milk. It does not have organisms in it, so it doesn't have a 2 week shelf life like pasteurized milk does. Uh, this slide is showing you that dry heat is effective at sterilization, but it is not as effective as wet heat. And this kitty is climbing in the oven to show you that dry heat is not as effective as wet heat. And you actually know this because you cannot put your hand through steam, boiling steam. You would be burnt, like immediately within a second or less. But you can put your hands in a hot oven, as the kitty is demonstrating. So dry heat is not as efficient as wet heat at killing microbes. Any question about any of that? Dry heat does kill organisms by oxidation. You can have flamming and incineration, which are so high heat that the cells are actually turned into char. But then there's baking, where you achieve hot air sterilization, but you need to put the object in the oven for 2 hours to get hot air sterilization, at least at 170 degrees Celsius, which I don't know what that is in cooking temperature. It's probably 300, 350 degrees. Let me go look that up. Ah, come on. Whoop. 307 degrees Fahrenheit. So it's a little warmer than I was saying. 370 degrees Fahrenheit. Requiring 2 hours. That's pretty warm. Filtration is a mechanism we have for control microbes. It removes microbes from liquids and gases by sending the liquid or the gas through a filter, and then the microbes are kept on this side of the filter. There's no microbes on that side of the filter. It's useful for sterilizing liquids and gases. It cannot sterilize, obviously, a solid object. There has to be a pore in the filter that keeps the microbes on this side of the filter. Some juices, Coors beer, vaccines are usually sterilized or often sterilized by filtration. Milk is usually not sterilized by filtration because the fat, as well as the large proteins in the milk, will clog the pores of the filter. And that's why milk is often pasteurized. We can put the object at low temperature to inhibit microbial growth. This does not kill most cells, but it does prevent most cells from growing. Refrigeration, putting the object between 0 and 7 degrees Celsius, most refrigerators are around 4 degrees Celsius, maybe up to 7 degrees Celsius, slowing the metabolic rate so that the microbe cannot reproduce or synthesize toxins. It does not prevent psychotrophs from growing. So psychotrophs can grow at about, they can divide about 3 times per day and can reach about 2 million cells within a week. And that's why food in your refrigerator, after it's been in there for about a week, you should throw it away because these psychotrophs can grow and then spoil the food. They won't produce a toxin, but they can spoil the food. So any food that doesn't have a preservative in it, like high sugar, high salt, high acetic acid, meaning a lot of acids, should not be in the refrigerator for more than about a week. Freezing stops metabolic activity. Freezing does not kill most microbes. So if you were to freeze most microbes and then thaw them, about 50% of the microbes will still be alive. But it does stop metabolic activity, so the microbes could not spoil the food. They will not reproduce. They will not have any microbe activity, and they'll also prevent the synthesis of toxins. A slow freezer is like your home freezer. it allows ice crystals to form, which will kill some bacteria, but not all bacteria. About 50% of bacteria cells will survive, but it will kill all parasitic eukaryotic species. So one thing you can do to kill any eukaryote is simply to freeze it in your freezer. That would not be fast freezing. That would be putting the object in liquid nitrogen. That would not kill eukaryotic species because you do not allow the ice crystals to form if you put the object in liquid nitrogen. So quick freezing in liquid nitrogen does not kill bacterial cells. They'll survive that, and it would not kill a eukaryotic species like the eggs to a worm, for example, or the cyst to a amoeba. But if you put it in this home freezer, you're slow freezing, you would kill those eukaryote parasites. Any question about low temperature? Now, the trouble is when you then remove the food out of the refrigerator or out of freezing, the microbes that are in it can then resume their growth. High pressure transfers pressure evenly throughout a liquid, killing vegetative bacterial cells. It would not kill the endospores, so the endospores could survive. High pressure denatures proteins, alters carbohydrates. Endospores are resistant and they will survive. But when you're using high pressure, you can preserve the color, the flavor, and the nutrient value of the food, like the juice. And some juices are put under high pressure and then capped and then sold. I think because that's under pressure and then capped, the endospores do not germinate until you open it. But the endospores would survive. Some juices treated with high pressure to preserve the color, the flavor, and the nutrient value. Other juices will be filtered. And then some juices are pasteurized, in which case it would have a shelf life. And then some juices are actually heated, which would obviously destroy the vitamins, could change the color and the flavor. Vitamin C, for example, if you heat it, even pasteurizing it, as well as sterilizing it by bringing it to high temperature, would kill the vitamin C. But the vitamin C would survive the filtration or the high pressure. And there's a little joke. 2 lumps of coal. Mother says to baby, your dad's been under a lot of pressure lately. Lump of coal changed to a diamond. Desiccation and osmotic pressure have been used by humans for centuries to help preserve food. Desiccation is simply drying the object. This prevents microbial metabolism, and cells can remain viable for years, even decades if they're endospores. Resistance to desiccation varies by species, like E. coli, would only survive a few days, if even that long, if you dry out the food. Examples of desiccation include dried fruit or dried figs. Osmotic pressure is something we also use, very similar to desiccation. Using high osmotic pressure, either a lot of salt or a lot of sugar, causing plasmolysis. Plasmolysis is when the water leaves a cell and then the cell membrane shrinks away from the cell wall, which for bacteria would be very effective at killing the cell. Osmotic pressure is used all the time in molasses and honey, salted fish, using high osmotic pressure to preserve the food. And as you know, a bottle of molasses can stay at room temperature for years because of the high osmotic pressure. When the microbe falls into it, plasmolysis will occur, and then the microbe will most likely die. If it's an endospore, it can survive, but it will not germinate. And so the molasses can stay at room temperature for years. If you remember, I mentioned earlier that my mother had a bottle of molasses that we figure was at least 30 years old. And it was still good, we know, for 20 years because I made molasses candy out of it and fed it to my father, and he enjoyed it. We didn't use the molasses for 10 years later, either I or my mother, and that's why she finally threw it away about 10 years later. Any question about desiccation or a high osmotic pressure? All right. So that's those physical methods. Let's talk about the physical method of radiation. Radiation damages DNA. Here we're looking at the electromagnetic spectrum, gamma rays, X-rays, UV light, visible light, infrared light, microwave, radio waves. Only gamma rays and X-rays, as well as UV light, is considered radiation that kills cells. Ionizing radiation, gamma rays and X-rays, and electron beams have a short wavelength and ionizes molecules in water, producing an ion, meaning the high intensity radiation has a lot of energy, which hits an atom, causing the electron to absorb that energy and giving that electron enough energy to leave the atom, creating the ion. The ion can then react with organic molecules, especially DNA, to mutate the DNA. Ionizing radiation is often used for sterilizing food, like if you have your camping food and there's liquid in it, it'll be probably ionized. Pharmaceuticals, medical supplies. UV light is one form of non-ionizing radiation. You should realize that visible light is also non-ionizing radiation, as well as everything higher in the spectrum. But UV light can damage DNA. When UV light hits DNA, two thymines, which are right next to each other, will absorb that UV light and then cause the thymine to make a dimer between them that are two covalent bonds that connect between the thymines. So it's not one chemical bond, it's actually two uh, covalent bonds. Thymine does not normally bind to thymine, but if you expose it to UV light, it'll get a dimer between the two thymines. It has to be two thymines that are right next to each other, and the DNA will form a bubble here because the dimer of thymines will not hydrogen bond to the corresponding A. This will foul up DNA replication as well as RNA transcription. That this region of the DNA cannot replicate, at least correctly, and cannot transcribe correctly. Any question about UV light and how it damages DNA? Microwave radiation is not particularly antimicrobial, but microwave does create heat. Usually the water molecules absorb the microwave and then cause the heat start moving around, and then the heat can kill microbes. Any question about how radiation kills microbes? All right, this summary is showing you the different mechanisms of physical methods for controlling heat, moist heat, boiling, flowing steam, mechanism of action. You don't really need to know that, but it's used for cleaning dishes, basins, pitchers, autoclaving, how we make our media sterile, very effective, killing even endospores, pasteurization, how we pasteurize milk, cream, certain other beverages like beer and wine, dry heat, direct flaming, incineration. You have so high heat, you actually turn the carbon in the cells to char. Hot air sterilization, like baking, killing things by oxidation, does require long periods of time, longer than wet heat. Where is that right here? So about 10 minutes, killing almost all vegetative bacteria, fungal pathogens, most viruses. And you need 2 hours time of hot air baking. Filtration, removing the microbes by a filter. Cooling, refrigeration, deep freezing, uh, stopping the microbes from growing, um, stopping the microbial action if it's anything but the microbes that grow in the refrigerator. If it's a mesophile, you stop the microbe from having metabolism, but refrigeration has a bacteriostatic effect. It doesn't necessarily kill the microbe. And the food or drug would be good for only about a week unless there's another preservative in it. Like a bottle of ketchup has both high salt, high sugar, and high acid, meaning a lot of acid in there. And so the bottle of ketchup can stay in your refrigerator for years. But something like a hamburger, which doesn't have high salt, high acid, high sugar, can only stay in the refrigerator for about a week. We didn't talk about lyophilization. You don't need to know about it. High pressure, altering the structure of proteins, carbohydrates. Something that's often used, fruit juices can be treated with pressure to preserve the color, the flavor, and the nutrient value. Desiccation, food preservation, like when you dry fruit. Osmotic pressure, food preservation, like molasses or honey. Radiation, ionizing radiation, sterilizing pharmaceuticals, medical supplies, non-ionizing radiation. I forgot to mention this. Non-ionizing radiation is not very penetrating, meaning it really only sterilizes the surface. So if you wanted to clean something deep inside, you would not expose it to UV light, you would expose it to ionizing radiation. But if you only want to kill the surface, then you could use UV light. And one thing we did sterilizing the goggles in our lab was we'd expose them to UV light, killing the surface microbes. And we weren't really interested in sterilizing inside the glasses. You're only interested in sterilizing the outside of the glasses. All right, enough of the physical method. Let's talk about the chemical methods for controlling microbes. You should first realize that few chemical agents achieve sterility. Most chemical agents only decrease microbe numbers to safe levels, like removing vegetative forms of a pathogen from an object or desiccating the toilet. You're not removing endospores at all. when you bleach the toilet, and but you are removing vegetative pathogens from the toilet when you're using like bleach on the toilet. Any question about any of that? So disinfection refers to the destruction of vegetative pathogens. There are different factors related to effective disinfection. The concentration of the disinfectant can apply or affect the disinfection. Generally, most agents, the more concentrated the disinfectant agent, then the better the disinfectant will be. There is an exception with that, and that is with alcohol, and that is 100% alcohol is not as effective as uh, 75% or 70% alcohol. So with alcohol, you get 70% or 75% alcohol. It's at maximum killing. You go higher, and it is not as effective at killing. But generally, the more concentrated the disinfectant, the better it will be killing. The time also is important for the disinfection. You get greater disinfection the longer you apply the disinfectant agent. The pH can affect the disinfection. Generally, the more acidic the environment, the greater the disinfection. But it is true that around pH 7, the least effective the disinfective agent will be. The presence of organic matter can affect the disinfection. Generally, organic matter can reduce the effectiveness of the disinfective agent. For example, bleach is highly inactivated by organic matter. And so if you're applying bleach and there's a lot of organic matter around, like a lot of vomit or a lot of manure, then you need to use a lot of bleach to kill off all of the microbial agents. And organic matter is usually found in clinical situations. There's different types of disinfectant. So these are the different families of disinfectants, the phenol, the phenolics, the biphenyls, the biguanides, the halogens, the alcohol, heavy metals, surface active agents, chemical food preservatives, antibiotics, aldehydes, gaseous chemosterilizers, and then peroxygens. Generally, chemical disinfectants are not effective at killing endospores. There is an exception. The peroxygens do kill endospores. And there's another one, I don't remember which one it is. I'll have to come up with this. You don't need to know this table, but you should know the chemical agent, like phenol. You don't need to know the mechanism of action. Phenol is actually used very rare today, but it was the first antiseptic used to make aseptic surgery. Phenolics, a compound related to phenol, which are used today to kill surfaces or sterilize surfaces, or I should say disinfect surfaces, instruments. Biphenyls are sometimes found in hand soaps, skin lotions. Bioguanides like chlorhexidine, skin disinfection, especially surgical scrubs. So somebody who said chlorhexidine was correct. Halogens, this would be iodine, tincture of iodine, and then chlorine bleach. Iodine used as an antiseptic, and pretty good antiseptic too. Chlorine bleach, generally used as a fairly good disinfective agent. Alcohols, this is a very weak either disinfectant or antiseptic. It's so weak, it's more often called a degerming agent. because it's so weak. Heavy metals, this use has come back because of the antibiotic resistance to microbes, to antibiotics. It's sometimes used like silver nitrate or some other heavy metal um, in a what do you call that? A dressing, hospital dressing, to prevent the microbes from growing, like in a wound. For a long time, heavy metal use was discontinued because there were better agents like antibiotics. And then the heavy metals are usually toxic to humans if you were to consume them. Heavy metals on the skin is okay because the skin doesn't absorb the heavy metals, at least not very rapidly. And so, but their use has come back because of antibiotic resistance. Surface active agents found in soaps and degerming agents, sanitization, sanitizers, chemical food preservatives. We pour organic acids like sorbic acid and benzoic acid in our food, cosmetics, shampoos, calcium propionate we put in bread to help preserve the food or the cosmetics, shampoos, nitrates and nitrites put in different meat products to help preserve the food. Aldehydes, very effective antimicrobials like formaldehyde, sometimes used as disinfecting medical equipment. Ethylene oxide, a very effective gaseous sterilizing agent. The trouble is it's very toxic to humans, so you wouldn't put it anywhere where somebody will breathe it in. But it does kill agents, but like endospores, it does kill them, but you need long-term exposure. Uh, Chemical sterilizers sometimes used on hospital equipment or something that's difficult to sterilize because it can't take heat. And then peroxygen using oxidation. It's a very effective antiseptic agent and it can kill endospores. And so this would be one of the chemical disinfective agents that can actually kill endospores. Any question about any of that? And then peroxygen is used as a good antiseptic or a good disinfectant, like hydrogen peroxide used mostly as an antiseptic, but you could use it on your toilet and would be used as a good disinfectant. This is comparing the effectiveness of various antiseptics. Tincture of iodine, 20-minute exposure kills about 60% of microbes. And when we apply tincture of iodine on this patient's skin, the tincture of iodine will remain on the skin longer than 80 seconds. And you can see after about 80 seconds, let's say 90 seconds, you would have the 90% of the microbes dying. After 90 seconds, and the tincture of iodine actually would be on the patient's skin for long periods of time because you mark it and then the surgeon can see where the tincture of iodine was and make sure he doesn't cut outside the area exposed. Comparing that to soap and water, after 90 seconds of exposure, you have less than 90% of the microbes dying. So soap and water is good at washing away microbes, but it is not good at killing microbes. With nobody washes their hand for 90 seconds, you might be washing your hands for about 30 seconds, and you'll kill less than 90%, probably less than 95%. I mean, less than 10% or less than 5% of the microbes if you're washing your hands for 30 seconds. So hand washing mainly is effective at simply washing away the microbes. It is not really killing microbes. Any question about any of that? All right. We have a little bit of time to talk about the microbial characteristics, and that is different microbes have different characteristics, and these characteristics come into playing in microbial control. This is a simplified hierarchy of the relative resistance of the major microbial groups to different chemical biocides. Realize that specific agents may act differently on specific species of microbes. The most resistant microbe to control is a prion. The only way we have for effectively controlling prions is to incinerate it at like 1,000 degrees, I think it's Fahrenheit or Celsius, to kill the prions. And there's a good question out, and that is, why does the prion protein require such high heat to be destroyed? And there's not a good answer there. The other effective way to control prions is you can apply proteinases. The proteinases will degrade the prion protein, and you can control the prions by applying proteinases. The next effective, most resistant microbial characteristic is if the microbe makes endospores. Endospores are really hardy and hard to kill. Mycobacterium would be the next most resistant, and the mycobacteria have a lot of mycolic acid in their cell wall. That mycolic acid helps the cell survive many different agents, including chemical disinfectants. If the chemical disinfectant cannot get through the mycolic acid, the chemical disinfectant will not be effective at killing mycobacteria. The cystiprotozoa, the next most resistant, vegetative protozoa, next most resistant, and finally we get a bacteria, the gram-negative bacteria, the next most resistant microbe, and it's more resistant than the gram-positive bacteria. That's because the gram-negative bacteria have an outer membrane on their cell wall that controls what gets into the cell. And if the chemical disinfectant cannot get through the gram-negative outer membrane, then that chemical disinfectant will not be effective on the gram-negative bacteria, but it will be effective on the gram-positive bacteria. And a classic case of something that the gram-negative bacteria are not sensitive to is penicillin G, because penicillin G cannot get through the outer membrane of gram-negative bacteria, but penicillin G can get through the gram-positive bacteria and then harm the gram-positive bacteria. Fungi, the next most resistant, including fungal spores, they're a little more resistant than fungi. Viruses without envelopes, next most resistant thing. Gram-positive bacteria, one of the least most resistant. So gram-positive bacteria is easier to kill than most other microbes. And then the easiest thing to kill will be a virus with a lipid envelope. They are easy to kill. The lipid envelope has to be intact. And with like COVID-19, with a day on paper or cardboard, the lipid envelope will dry out, and then the COVID-19 won't be infective. It does take longer on plastic or metal. It may take 3 days. So this is the simplified hierarchy with the most resistant up here and then the least resistant down here. Any questions about any of that? I've already mentioned that biocides tend to be more effective on the gram-positive bacteria than the gram-negative because of the outer membrane on the cell wall. The boron protein can be selective on what it allows into the cell. The mycobacteria also very selective. If the chemical disinfectant can't get through the mycolic acid, it won't be effective on the mycobacteria. And then I've already talked about the endospores. The endospores are very few chemical disinfectants which can get through the endospore and kill the endospore. One exception is the peroxygens like hydrogen peroxide. It does kill endospores. I already mentioned about the viruses, prions. How do we kill them? Not completely solved yet. You can incinerate the cattle, the dead cow here, or the dying cow, and that's how the British got rid of their cattle. They incinerated them. But you can use for like surgical instruments. You can treat them with proteinases to remove prions. All right, biocides are not uniformly effective against all microbes. The compounds discussed in this lecture are not generally useful in the treatment of disease, with the exception of the antibiotics. And then let's talk about something else here. The mycobacteria are a microbe that are highly resistant to a number of different chemical disinfectives, but the characteristic of the mycobacteria makes them very sensitive to alcohol, like rubbing alcohol or ethanol. This alcohol can get right through the mycolic acid, and then that makes the mycobacteria highly sensitive to the alcohol. So the mycobacteria, even though it tends to be resistant to most of the chemical disinfectants, that same mycolic acid makes the bacteria highly sensitive to alcohol. Any question about how the different characteristics can affect microbial control? All right, if not, I'm done with this lesson. Any questions about Chapter 7? Remember the worksheet on Chapter 7 is for extra credit, and it is due 11:59 p.m. Thursday, 6/12. Let me also remind you, your unknown project will be due 11:59 p.m. this Saturday, June 14th. Your unknown notes pages will be closing Friday, June 13th at 11:59 p.m. And if you request something on Friday, it must be a test that will take a day or less to get the results. Oh, and I should remind you also, the unknown notes pages will be graded. You should finish your unknown notes pages where you state your last results. A lot of you have not finished your unknown notes pages, and you should finish it because this assignment will be graded. Any questions about any of that? Any question about anything? All right, I'll be in the lab at 6:30 to 6:45. to answer any questions. If you don't have any questions, you don't need to show up in the lab. All right, bye.